Okay. We'll get a little bit on this. First of all, what's an allele? Right. It's a version of a gene. So instead of saying the version you got from mom or dad, you actually say we got an allele from mom or an allele from dad. This is kind of a review. Which of the patterns can't belong to the child of this couple? Let's see. So let's see. Which one? I'm looking at it. Okay, so let's see. This one, B? D. Yeah. Okay, so they got that from mom. No, and that from dad, so that's not D. That from mom.
Yeah, because C is a complete repeat of mother.
Yeah, right. So it's C, right? Mom and mom. Can't get two alleles from mom. Okay. Which cannot be the child of this couple?
Yeah, B. Because it's the same thing as the previous one.
Mm-hmm.
Copy mom.
B?
B.
D. Yeah.
Oh, okay. I didn't notice that one.
That's a double.
So it's B, C, D.
And dad and mom, see this one is okay because they both have the same allele, right? This one is mom. Hold it.
No, never mind. Because dad is also.
That's dad and that's mom, right? So yeah, so it's got to be mom and dad. This one, right? Okay. So what kind of cell is this? Eukarya or bacteria or archaea? Huh? Eukaryote, right. Because it's got a nucleus and all these other organelles. So who are these guys? Right. Oh, by the way, they got a Nobel Prize for that. So what's the complementary for this? Which one for this? G, C, C,
A, T.
right? Do that. Idiot. Okay. It turns out 2% of the genome of those 3,000 base pairs actually codes for a protein. And you might think, well, what does the other millions and billions of base pairs actually do? Well, for a long time, it was thought that one hypothesis was that it was junk. It was accumulated over billions of years of evolution, and a lot of it doesn't do much of anything. There's these things called pseudogenes that look like genes, but they've got stop codons in the middle of them, so that it doesn't look like they're functional. And then there's also what are called transposable elements. These are considered parasitic pieces of DNA. What they do is they have their own DNA polymerase, and they copy themselves into the genome all over the place. And our genome actually has a number of mechanisms to control this copying. Eventually, though, a lot of those transposable elements actually get assimilated into the genome, and it looks like at least 20% of the genome is based on regulatory elements. That means gene regulation. And that's what we're going to learn about today. So we know about, let's say, a protein like lactase, which is an enzyme. Remember, ASE is an enzyme, and it helps us digest milk. And people who are, let's say, lactose intolerant, they can't digest milk. They can't break down that disaccharide. What's a disaccharide? Anybody? Huh? Okay, well, lactose is a disaccharide. So what is it made of? Huh? No, no protein. It's actually a sugar, okay, like a carbohydrate. And it's made up of a glucose molecule and a galactose molecule. And to actually get it to cleave it so that you end up only with a glucose molecule or galactose molecule, you need a certain enzyme to do it. Now, we've talked about this a little bit before. Obviously, mammals make milk, and most mammals, after they're weaned from their mother, they never come across milk for the rest of their lives. Maybe they're, a house cat or something, but they can't digest it either. So it's weird that there's actually humans that can digest lactose as adults. And because making lactase, the enzyme, is expensive. Why would you want it to be on when it's no longer needed for adult mammals, right? Well, there was not obviously, but there was a mutation. In fact, at least 3 different populations have similar mutations that keep the lactase gene on. And those people can digest milk, and the people that don't have it can't. And it's usually associated with certain populations that were pastoral. That means they had animals like goats and sheep and cattle that they could actually milk. Now, someone like that has a real advantage. If they can have a food source like that and no one else can, and so you'd expect, what would happen in terms of evolution? Would that mutation increase or decrease in a human population? Increase, right. So for those populations, almost everybody has this mutation and they can drink milk. There are other populations of humans that never had a pastoralist culture. They didn't do animal husbandry. And so usually those different populations are the ones that are lactose intolerant. Okay, now, so lactose or lactase is the enzyme. That is a protein. And it's composed of a whole string of codons, which code for certain amino acids. Okay. Okay. DNA polymerase. The double-stranded molecule unwinds an enzyme called DNA polymerase, then adds complementary nucleotides. So, in other words, each of these strands is a parent strand, and then a new strand, when the cell divides, complements the base pairs, right? So, let's say, this one will have to be this one, the complement, right? So what it does is it actually starts adding on, let's say the parent strand is, okay, there we go. So here's the parental strands, right, along here, and then on the inside is the complementary base. Okay. What procedure will a forensic scientist do? We already know that. PCR, amplify it. Okay. You can read this yourself, but you can get a genetic profile with a number of repeats. Remember how this goes? So larger fragments move more slowly. Smaller fragments move more quickly because they're lighter. Okay, so here's a genetic code. Like I said, there's 20 amino acids. If you look at these letters, like TAT, that codes for tyrosine, which is an amino acid. You need 3 letters to make an amino acid, or a code for a certain amino acid. It's always, a codon is always 3 letters. Now, if you're using the transcript, which is made of RNA, then you actually, instead of using a T, you use a U. It's called uracil, but that's besides the point. Okay, so there's 20 amino acids, 3 base pairs long, code for an amino acid. Some of these are actually redundant, and then some actually code for what are called stop codons. This is genetic code, and as you can see, there are always three letters of the genetic code for one of the 20 amino acids. And this is phenylalanine. Okay. TTC also does phenylalanine. It's somewhat redundant. Usually in the 3rd base pair, you end up with an identical amino acid. There's 20 of them, and obviously you get more with this. There's also some amino acids like this one that are stop codons, and that means they stop the transcription and translation of the code, genetic code, into a protein. So, now, it turns out, which we're going to learn in a little second, you wonder, well, what's UUU? Well, messenger RNA, so the DNA is transcribed into messenger RNA. We're going to look at that in 1 second. But, and messenger RNA has one difference, and that is it uses a U instead of a T. And messenger RNA is a single-stranded instead of double-stranded. Okay. That's good. Well, I think next week is a test, so we're going to have to pick up where
That's not right.
Next week?
I think it is, right?
It says the 23rd, but Friday's the 21st.
What?
It says the 23rd, but Friday's the 21st.
Okay.
I'm assuming.
Yeah. That seems so soon.
Yeah, because we got a test last week.
Yeah. I don't know. Would you like, maybe we should like, would it be okay if we slipped it down to the next Friday?
Sure.
Yeah, I'll send an email and I'll see how people feel about it.
Half the people don't even know.
Take your sweet time, I got nothing to do.
So, DNA is basically, the chromosome is made up of DNA. And we're going to look at how it's made, what it consists of. But on each chromosome, there are a whole bunch of genes. It used to be thought that much of the genome, like I said, was junk, but now it's not so much. There are less than 20,000 genes that make mostly proteins and some different kinds of RNA. And then the proteins are the things, remember, that actually do things, right? They do all the work. Okay. Now, so each of these amino acids, they're all different, or there's the same, but they're strung together in various orders. So this is glycine. I'm not sure what that one is. Yaline, glyn, there's 20 of them. They're strung together in long polymers, just like carbohydrates are, just like fats are. Except what they have is they have an amine end, but you don't have to know that. Okay. Let's see. What's a complement to AGA? T, what's next? C? T. Okay. Does everybody understand that? Okay. Who doesn't understand that? Okay. Let's see. Let me go back. Now, for a gene to actually make a protein, there's 3 steps. That's 1. Okay. So, the first step is that the DNA is turned into an RNA that is the exact complement of the DNA. So the strands open up, and one strand is then copied into an RNA, with the U's replacing the T's. That's messenger RNA, which is then carried out into the cytoplasm, which we're going to look at now. Okay, so here we are. Synthesis, Now remember, all this happens, the first part all happens in the nucleus. This is all the nucleus. And here's the DNA, all right? Okay, and then here's a complement of messenger RNA. And that is then ferried out through a pore, which is another protein, into the cytoplasm. And in the cytoplasm, that RNA is actually red, and it is red in this thing. So this is the RNA, right? By this ribosome, and then translated into amino acids, which are then the proteins. And it reads the code. Each codon is 3 DNA or RNA letters, and they code for a specific amino acid. So, DNA transcription to RNA. RNA is then, after it comes out into the cytoplasm and hooks up with the ribosome, it's translated into an actual protein. Okay. Now, if you remember, does a muscle cell have the same genes as a neuron? No, right? Except you're all wrong. Each cell has the whole genome, all of it. Doesn't necessarily use it, but it's all there. All 3,243,000,000 base pairs of it. And it's there in the form of chromosomes. Most of the time you can't see them, but when you replicate a cell, you can see them. And a muscle cell has the full genome. A neuron has full genome. The retina, the cones, and the rods, they have, each one has a full genome. The only difference is that they may use different genes and make different proteins. So, now a genome is really, well, it makes it sound like it's like a blueprint, or it's not really that. As you remember, genes are always found in the context of the environment that they're in. So it's actually, you can't just take this instruction book, make a bunch of proteins, and think all is good. It has to have some regulatory elements, depending on what the environment is, whether it's the outside environment, the cellular environment. Okay. Have one complete copy of the genome. That means 23 chromosomes. One from your mother and one from your father. Okay, now, so to make a whole human, now by the way, these are called, let's see, Okay. The gametes are haploid. They only have one copy, whether it was from the mother of, let's say, the mother's mother or father. And the sperm has one full set of chromosomes from the father, right? And then when you have a fertilized egg, they come together and suddenly you've got a diploid organism with two copies of each genome. And that's called, those are homologous genes. So, okay. Okay. PM, Papa, Mama. So you have one piece of DNA, a chromosome, with the exact same genes. And this is called a homologous pair. So homologous pairs are always paired up unless there's a problem during replication. Okay, now here we are with a muscle cell, and we've got genes A, B, C, and D. And these genes code for the muscle and the different functions that it does. Okay, so with gene A, you have transcription into RNA and then translation into protein A. There's, in this box, there's 25 circles, and that means 25 molecules of the protein. Now, go to the next. It also, gene B is transcribed and then translated into protein B, but you can see there's much less of it that's actually transcribed and translated. C, more than B, but less than A. And you've got protein C, and that's 13 base pairs long. And then, in this case, gene D is not turned on. It's not expressed. Okay. Okay, you can see that gene A is actually transcribed and translated the same in both the muscle cell and the neuron. Gene B is not expressed. C is transcribed and translated a little less than C in the muscle cell. And then gene D is expressed as well. Now, there are some genes in the cellular process that are always expressed. They're called housekeeping genes. So a cell needs to do cellular respiration, where they get ATP from the food. They turn glucose into ATP, which we went over a little bit earlier. And ATP is what the cell uses to actually drive chemical reactions and all the other things the cell does. So some of those genes are always going to be turned on and making protein. Then there will also be certain genes that are only expressed in that specific kind of cell. So in neurons, there is no B protein. There is a little bit of B protein in the muscle cell. There's muscle is D is turned off, and there's a fair amount of transcription and translation for gene D. So you get the idea that each cell has a complete genome. Some genes are always, whether it's a muscle or a neuron, are always turned on to a certain extent. Or, and then there's others that are specific to each kind of cell. Now, each DNA sequence each gene has DNA sequences that actually control gene expression. In other words, turning them on or turning them off. Promoters are actually elements that are just upstream of right next to the gene. In fact, that's where the mutation for galactase or lactase that can break down milk, that's where the mutation was that kept the lactase gene making lactase. There are also enhancers that can be on completely other chromosomes that also help regulate gene expression. So, does the promoter region or the enhancer act as an on-off switch? No. When someone says, oh, gene's turned on, it's not like that. It's a matter of how much it's turned on. So the whole system works more like a dimmer or a rheostat. If you go this way, you get more and more and more gene expression. Same thing with here, right? Gets warmer and warmer, more and more electricity. In this case, more and more genes. So it's not an on-off switch. It's an on switch that you can control specifically in terms of gene products. And then the coding region, of course, is for lactase. It's the lactase gene that makes the protein. But there's all these other elements that control whether cells make a specific protein or not. Okay. Now, let's take a look at this. These regulatory elements, they can be either promoters or enhancers. And you have these different organisms, human, chimp, mouse. The liver is expressed with these group of promoters or enhancers. The heart with these. And then the brain with these. And then all three species, you can see the blue, they're all using the same gene. In fact, actually, there's probably, very similar gene expression for all three. They're all mammals. They're all closely related to each other. You can see where certain substitutions happened. In the human, it's TGA-GGG. Its chimp is TGA-TTA. And then it's TGA-TGG, or GC. Okay, so there's been some mutations in this region in terms of, this is the regulatory element region. Okay, red, brown, green peaks are common to all, are identified by the open chromatin, histone modification, or transcription factor. The yellow peaks, almost all the genes that we have, well, they all were pretty much inherited from our other previous species. Like we're most closely related to chimpanzees. So we share more in common. But basically, and then there's some new ones for humans, obviously, that were made. But usually it's just a matter of expression more than anything else. These are some regulatory elements that are actually located far from the gene that they control. And this has been part of the genome. You know, first there was a genome project to figure out, get the full genome of a human sequence and all that. Well, now we're trying to figure out not only what genes are expressed in terms of proteins, but what elements are involved in that expression. And so they do a bunch of molecular biology techniques. They do something called assays to see which genes are actually expressing, doing something functional that then lights up with a corresponding gene that makes a protein. And then in C, you get a compilation of all the regulatory elements, connections to the target genes, you get a network of regulatory interactions that is unique for each tissue. When we saw the lactase gene, this whole lactase it's a complex, a 3D complex, with specific sites that are active sites. And it only works if it's in that right configuration. So here's the lactase gene. Okay. Lactase gene. This. And this is lactase. And this is lactose, which is a disaccharide. And it is then separated into two different kinds of molecules, glucose and galactose. Okay? So, now, what this does is it breaks, it's an enzyme, so it's a catalytic reaction. Normally, like I said, it's not turned on in adult animals, adult mammals. There's also a really good video on the evolution of lactose tolerance, where you can actually digest milk, where it happened, kind of how it happened in terms of evolutionary, in terms of evolution of it. It's happened in the last, probably 12,000 years of human existence. And humans, the first humans were about 200,000 years ago, if you remember correctly. Okay, so now when transcription actually happens in the nucleus, and there's this other protein, other enzyme called RNA polymerase that down here that adds these RNA nucleotides based on what the code is in terms of what these genes are, or what these codons are. So it puts all these nucleotides together that correspond exactly to the genetic code for DNA. Of course, the RNA that is translated or transcribed into is then ferried out of the cell into the cytoplasm, and it's translated using these ribosomes, which are these big protein complexes. And then in eukaryotic cells, these proteins are usually go through other processes that, like post-translational differences, that might be tweaking the protein. So now proteins have to be in the right conformation, like a 3D visual. It's not like they're just strung out in a long line. So when we saw the lactase gene, let's go back. Okay. God, what a mess. Okay, here we go again. That's mRNA. This is T. Oh, there it goes. RNA. And what the tRNA does is it attaches to a specific amino acid. Or rather, it already has an amino acid. So each of these are floating around in the cytoplasm, and the actual, the actual ribosome then reads the messenger RNA and puts the right amino acid in the protein. And of course, this is the amino acid chain that's produced. And it'll finally reach a point where a stop codon in the protein has been made. So, okay, good. So that's kind of the process of it. Okay. So the tRNA carries an amino acid. In this case, this one, phenylalanine. So here's the codon. This is the mRNA, right? It's got UUU, and then its anticodon, the complementary pair, is AAA. And then there's this region of internal complementarity, and if it's the right binding site, then you'll have the right amino acid. And this is how the whole complex kind of works. Remember, the codon is always a 3-letter code, whether it's in DNA or RNA. Okay. And here's the actual genetic code. As we saw before, the amino acids, as you remember, there are about seven essential amino acids, 3 that are kind of semi-essential that we have to get from our diet, and the rest we make ourselves. Okay. Does anyone in here, let's see, what is it called? It's escaped me. Oh, it's found, he's a Mexican, it's an herb. It'll come to me. Anyway, there are some people that can taste certain things and other people that can't, or they taste bad. The whole taste thing is a matter of an amino acid sequence that's coded into and turned into a protein. And these taste buds, these individual molecules, take a look. Okay. Okay. So, these individual molecules actually affect our taste. Now, the things that we like, probably other animals don't, or they usually don't come across in their diet. Or some animals come across things that we don't like vultures eat carrion. That means decaying flesh, right? It's just crawling with bacteria. If we ate it, we would probably get really sick. Also, it wouldn't taste good. It would smell, and it would smell just horrible, right? Carrion smells really bad. That tells us that we don't want to eat this thing. A vulture, of course, would smell that and go, it's dinner time, right? So it uses the different receptors in terms of what it tells, what the taste is. It goes for it. Now, it would like, if let's say it ate, let's say there was sugar, it would probably taste bad to the vulture. Okay. Now there's these different tastes. There's also another one that's called savory. And those are the basic tastes that we have. Bitter, we usually associate with some sort of toxin. So, like bitter almond, the almonds that we eat are sweet. They don't have cyanide in them, but there's a cyanide compound in wild almonds. And when we taste it, it tastes very bitter. It tells us, in fact, that this is not something that you generally want to eat. Okay. Now, this is a red blood cell. A normal looking red blood cell. This one's a little bit different. And then this one is really different. And a normal red blood cell It's made of hemoglobin, which is a protein that carries oxygen. There are some other creatures that use a copper-based system instead of an iron-based system. Hemoglobin is mainly made out of iron. And this is kind of the structure of it. It's on chromosome 11. There are, well, we'll talk about that in a second, but now, anybody know what sickle cell anemia is? Yeah? No? Okay. Well, I'll say it anyway. Sickle cell anemia is a genetic disease, right? It's a recessive genetic disease. And so if we actually look at, let me ask. Okay. And you can see where the substitution was in terms of the amino acid substitution. Okay, so the A was replaced with a T. Now, people who have sickle cell disease, there's, we'll say, one of the alleles, HBA, is the normal allele. And we have basically normal adult hemoglobin, and it looks like a certain way. This is the whole amino acid sequence and everything. People with this S allele make a deformed protein. It's only expressed in recessive, that means 2 copies. So people with 2 copies actually are the one that express sickle cell anemia. People that have one of each, they're called heterozygous. They don't get the kind of sickling that you see here. They get a somewhat different, it's pretty much a wild type, like you get just from the normal sequence. Not entirely, though. But it makes them resistant to malaria. The malaria parasite cannot get into the red blood cell, which is part of its life cycle. And so those people are resistant to getting malaria. And that's great, right? The thing is, obviously, with this recessive allele, if you have 2 copies, you get the disease, and those people have 0 fitness. So you're sacrificing a whole bunch of your children if they happen to get 2 copies of the S allele. That's like, can be 25%, let's say, or whatever it is. And those that actually have, that are heterozygous between, HBA and HBS versus HBA, those individuals are the ones that are resistant to malaria. So malaria has to be endemic. That means in an area, there's lots of malaria. There's lots of the Anopheles mosquitoes that carry malaria, and they go from one individual to another and bite and transfer malaria parasite. So this is a mutation that in some environments is actually beneficial, but only in the heterozygous form, HBA and HBS. HBA, when there's a lot of malaria around, all those individuals are susceptible to malaria. A lot of them get very sick, a lot die. Of course, individuals, so they're wiped out, so there's a real strong selection for the heterozygote. The homozygote of HBS, of course, is the lethal variant. That is the cost of actually having this resistance to malaria parasite. Oh, there's another thing. It turns out actually that the heterozygote does not carry oxygen quite as well as the normal HBA genotype. Okay, so those individuals, they carry oxygen almost as well. The cells are, red blood cells are a little misformed, but they're not enough to really stop what they normally do. In sickle cell, with those kinds of red blood cells, they get stuck in the capillaries, and they build up, and it's very, very painful disease. They're working on a cure, actually. They might have something pretty soon. Okay. Let's take a break.